Hello, Fernandez Rina. Are you there?
Yes, sir.
Hello. Yes. Am I audible?
Yes, sir.
Yes, sir. Okay. Good morning, one and all. Okay, shall we start? Okay, well, from microbial biodiversity, the unit which was assigned to me, that is domain eukaryon. And in that domain eukaryon, we have seen the molds or fungi. And in molds, we have seen fungi, the general characteristics, and one of the most important group of this mold, but it is separated from this fungi, which is yeast. And this yeast today would like to see in short. As all of you are aware, yeast mainly used for baking purpose as well as in brewing industry. Therefore, it is necessary you have to study the general characteristic properties and its a scheme of classification, because industrial point of view, it has got a lot of significance. So when you will go through this scientific literature, you will find, according to scientist Alexopoulos, the yeast is, they have categorized or they have put into a division a mycota and subdivision Eumycotonia, class Ascomycetes, subclass Hemiascomycetidae, order Endomycetales, family Saccharomyces, and species is a Cerevisiae. So all of you are aware that the yeast means immediately on your tip of tongue, yeast, example Saccharomyces cerevisiae, and this Saccharomyces cerevisiae yeast, which is used in baking industry, we call it as a baking industry, a baking strain of Saccharomyces. In brewing industry also it is used, in wine industry also it is used, as well as in certain other industries this Saccharomyces cerevisiae is used. So here you will find its what you can say this cell structure which is seen under electron microscope. You will find that the outermost layer cell wall is there. Cell wall is there. Then this is the mitochondrion is there. This is the cytoplasm. This is the nuclear membrane. Then this is the mitochondrion. These are the storage granules. This is the vacuolar membrane, vacuolar granules. Then this is a vacuole. Then you can see this is the nucleus. This is a nuclear core. And this is then storage granules. This is in short. This is in short cell structure. हे सेल स्ट्रक्चर जे आहे, आता तुमचा जो काय बदललेला पॅटर्न आहे त्याच्यामध्ये काय विचारलेलं आहे, 2 मार्काचे प्रश्न विचारायचे, 4 मार्क्सचे प्रश्न विचारायचे आणि 6 मार्क्सचा एक प्रश्न विचारायचा. देर इज अ चान्स, देर इज अ पॉसिबिलिटी, ड्रॉ अ सेल स्ट्रक्चर ऑफ ईस्ट सेल स्ट्रक्चर. अंडर दॅट सर्कमस्टन्सेस हे 4 मार्क्सला विचारलं जाऊ शकते किंवा 4 मार्क्ससाठी हे सफिशियंट आहे. त्याच्यामध्ये असे हे जे काय वेल डिफरन्शिएटेड सेल ऑर्गॅनेलिज आहेत, हे माइटोकॉन्ड्रिया आहे, न्यूक्लियस आहे, न्यूक्लिअर मेम्ब्रेन आहे, न्यूक्लिअर कोर आहे, न्यूक्लियस आहे, असे जे काय वेल डेव्हलप्ड or well differentiated cell organelles. Say for example, this is what you can say, this is a vacuole is there. Then in this vacuole, vacuolar granules are there. This is vacuolar membrane is there. हे मी कशासाठी सांगतोय की आता आपलं बदललेलं स्वरूप आहे, म्हणजे आता बदललेल्या स्वरूपानुसार आपणाला 4 मार्क्ससाठी काही प्रश्न विचारायचे. त्या 4 मार्क्ससाठी अशा पद्धतीने प्रश्न विचारला जाऊ शकतोय. Draw neat label diagram of the yeast cell structure. Yeast cell structure. Under that circumstances, you should be able to show the yeast cell structure. असं हे 4 मार्क्सला विचारलं जाऊ शकते. त्यासाठी तुम्ही त्या दृष्टिकोनातून पाहणं गरजेचं आहे. How many of you are there? 24. बाळांनो समजतंय का मी काय सांगतोय ते? दिग्विजय कुलकर्णी, आर यू देअर?
येस सर.
येस सर. तर ते बदललेलं स्वरूप आहे, त्या बदललेल्या स्वरूपानुसार वेगवेगळ्या प्रकारचे 2 मार्क्स, 4 मार्क्स आणि 6 मार्क्सचे प्रश्न विचारायचे तुम्हाला. आणि ते 2, 4 आणि 6 मार्क्सचे प्रश्न विचारत असताना डायग्राम सुद्धा विचारले जाण्याची शक्यता आहे. त्यासाठी मी आता तुम्हाला Then next, silent features of the yeast cell. As far as silent features of the yeast cell is considered, one of the most important silent feature of the or important characteristic is they are unicellular. Though yeast is a eukaryotic, most of the eukaryotic organelles are multicellular in nature, whereas the yeast is a unicellular eukaryotic in nature. Unicellular eukaryotic. And next, yeast cells are non-mycelial. It is as I have told you people, in broad sense, it is called as a mold or it is called as a fungi. But to be more specific, it is called as a yeast, and this yeast is a non-mycelial. So in case of most of the fungi and molds, you will find mycelia thread-like structure, whereas it is a non-mycelial saprobic fungi. That saprobic fungi means they break down this dead organic matter, therefore it is called as a saprobic fungi. They do not form a spore fruit, whereas in case of most of the fungi, they produces the spore, various kinds of or different kinds of the spores, whereas in case of this yeast, they do not form a spore fruit. The yeasts are degenerate, that is, and they lack properties like mold, therefore it is called as a degenerate, that is they become less specialized. That is, in case of eukaryotic single-celled members, which is called, we call it as a fungi kingdom. Whereas in case of degenerate, that is, they lack properties like mold. Therefore, it is called as a degenerate. Whereas, as far as their habitat or occurrence is considered, they are found in nature, in liquid organic material, in soil, in animal excreta, and on surfaces of ripe fruits and nectar of flowers where sugar percentage is there. So you might have been observed a white layer at the white layer on the grapes, white layer on the apples. त्याचे जे काय देठाच्या बाजूला व्हाईट लेअर असतं ते कशाचं असते ते ईस्टच असते देठाच्या बाजूला जे काय व्हाईट लेअर आपण पाहतो. Then the next silent features, they are capable of one of the most important characteristic property.
वेटिंगला मुलं आहेत.
Yes.
वेटिंगला मुलं आहेत सर बाहेर.
ऍडमिट केलेलं आहे ऑल सगळ्यांना रे बाळा. Yes, anybody is having problem? I have admitted all. Yes. Nihal Mullah. 25 जण जॉईन झालेले आहेत. 25 स्टुडंट्स आर देयर. Then the next important general characteristic property or silent feature, that is, the yeasts are capable of fermenting carbohydrate solution producing ethyl alcohol and carbon dioxide. One of the most important desirable characteristic property of the yeast, which is exploited in the industry, that is, they have capacity or ability to ferment various carbohydrates into ethyl alcohol and carbon dioxide, and this ethyl alcohol is mainly used for various purposes. It is mainly used as energy purpose. तर ते इथाइल अल्कोहोल प्रोडक्शन साठी मेनली इंडस्ट्रीमध्ये ईस्ट वापर. The thallus is a minute hyaline structure. The thallus is a what you can say, this minute hyaline that is colorless or transparent oval or spherical cell, which is differentiated into distinct cell wall enclosing the protoplast. Distinct cell wall enclosing, this is the protoplast. Cell wall च्या आतमध्ये काय आहे, protoplast आहे. The cell wall is a thin, which is a delicate and chitinous in nature. Cell protoplast consists of granular dense cytoplasm, a minute nucleus, and large vacuole and mitochondria. Large vacuole and mitochondria. The vegetative reproduction takes place mainly by budding. In some yeast, it takes place by method of fusion, that is what you can say, this union of two yeast cells, that is a fusion. You can say this division or splitting into two. It is a form of a nuclear division or binary fission, and budding are the two common methods of asexual reproduction. Asexual reproduction. So, ascospore formation is the usual method of reproduction. Their formation involves sexual spores. The enlarged diploid old yeast cells under adverse circumstances function as an asci, and the diploid nucleus of the asci cell undergoes meiosis to form, that is a reduction division, to form a 4 haploid nuclei, which are organized into non-motile meiospores known as ascospores. The mature ascus thus contains 4 to 8 ascospores, and the liberated ascospores under favorable conditions germinate, and each produces small haploid spherical yeast cell. Here and in the next slide, it is shown schematically or diagrammatically. It is shown in the next slide. This is its ascospores formation is the usual method of reproduction, and their formation involves what you can say this sexual process and this enlarged diploid old yeast cells under adverse circumstances which functions as an assaye. This is a total assaye, and in that assaye these are the ascospores are there. These are the ascospores. This is the assaye. Then Saccharomycete. So this family includes a group of reduced degenerate ascomycetes, popularly called as yeast. So they are non-mycelial ascomycetes that form no fruiting bodies. Their usual dominant growth form is unicellular, which may be what you can say this, which may be globose, ellipsoid, or shortly cylindrical. Globose, that is having what you can say having shape of a spear or a ball shaped sphere, is ellipsoid, elliptical, or dumbbell shape, uh, ellipsoid. So Saccharomyces cerevisiae, where ellipsoidans, which is used for wine production. Saccharomyces cerevisiae, where that is its variant or its strain ellipsoidans. तो elliptical त्याचा स्ट्रक्चर असते. एक काय होतं असतं आपल्याला मायक्रोस्कोप मध्ये अंडर पाहिलं आपण मायक्रोस्कोप खाली तर त्याचं जे काय स्ट्रक्चर आहे ते ग्लोबोज इलिप्सॉइड और शॉर्टली सिलेंड्रिकल इन द फॉर्म आपल्याला ते वेगवेगळ्या प्रकारचं स्ट्रक्चर ओळखता येत असतं किंवा दिसत असतं पाहता येत असतं. Saccharomyces cerevisiae, a diploid yeast cell showing a structure. That is, this is the diploid yeast cell structure. Haploid and diploid, these are the mating types of the yeast. Mating types of the yeast. Cell wall, cytoplasm, darkly stained, etc. Then yeast have following characteristic property. The thallus is a non-mycelial, predominantly unicellular structure. It reproduces asexually, either by budding or by fission, sometimes by both methods. The zygote, the zygote, that is the gamete which is formed by union or fertilization event between two gametes, that is which occurs mostly in eukaryotic cell. The zygote, which is a result of fusion of two cells or develops parenthogenetically, develops so what you can parenthogenetically, that is reproduction from an ovum without fertilization, reproduction from ovum without fertilization, parenthogenetically from vegetative cell, and functions directly as an ascus and produces ascospores, and the ascus is naked. Most of yeast have ability to ferment certain sugars. Most of the yeast have ability to ferment certain sugars. This is one of the most important desirable characteristic property of the yeast. As far as its habitats or occurrence is considered, is they are present um, everywhere on the face of earth. They are widely spread in distribution. Widely spread in distribution. They are present everywhere on the face of earth. They are saprophytes. They can grow on dead decaying organic matter. In nature, the yeast occur in the sticky sugary exudate on the surfaces of sweet fruits such as apples, grapes, dates, etc. Because for the growth, basically they require a sugar percentage. So on the surfaces of apples, grapes, dates, yeast can grow. They are abundant in vineyards and orchards. Few species of yeast are parasitic to human being, plants, and animals. Some of the species of the yeast are pathogenic to human being, plants, and animals, and they can cause various infections or diseases. As far as types of yeast or kinds of yeast are considered on the basis of asexual method of reproduction, yeast are roughly divided into three categories. That is what we can say this. That is first category. That is a budding yeast. So this budding yeast which reproduce asexually by the method of budding, in which a parent cell buds off a small daughter cell called the bud. And the most common example of budding is are the genus Saccharomyces cerevisiae. Saccharomyces cerevisiae again, Saccharomyces cerevisiae again, which is used in brewing industry and baking industry. The brewing strain and baking strain it is called as, and Saccharomyces ellipsoidans, which is used in wine industry. Wine industry. Budding yeast. Saccharomyces cerevisiae showing vegetative reproduction by budding. हे vegetative reproduction by budding. आता जे काय मी व्हेजिटेटिव्ह रिप्रोडक्शन बाय बडिंग दाखवली, ही 2 मार्क्सला सुद्धा डायग्राम विचारली जाऊ शकते. ही डायग्राम 2 मार्क्सला सुद्धा विचारली जाऊ शकते. ड्रॉ बडिंग येस्ट. तर आता आपलं पेपरचं जे काय स्वरूप बदललेलं आहे, नेचर बदललेलं आहे, त्याप्रमाणे आपल्याला सामोरं जावं लागेल. 2 मार्क्सला कशा पद्धतीचा प्रश्न विचारला जाईल, 4 मार्क्ससाठी कोणत्या पद्धतीने, 6 मार्क्ससाठी कोणत्या पद्धतीचा प्रश्न विचारला जाईल. त्यासाठी आत्ताची काय मी डायग्राम दाखवली बाळा, ती विचारली जाऊ शकते. Saccharomyces cerevisiae showing vegetative reproduction by budding. मयुरी जाधव आहे का? मयुरी जाधव
हो सर.
आहे. अरे कुठे बाहेर आवाज येतो?
अहो सर बसमध्ये मलाच ऐकायला येत नाही.
बस मध्ये आहे.
हो.
अरे then next to this budding is diagram. E to C सगळ्या डायग्राम्स आहेत, त्याच्यातली कुठली 2 मार्क्ससाठी काढू शकता. त्यानंतर फिजन yeast. त्याच्यानंतर फिजन yeast, that is they reproduce asexually, in which parent cell divides by transverse division into two daughter cells of equal size, and the process is called as a fission. And the best example of fission is a Schizosaccharomyces, and it includes 3 species. Schizooctoporus is well known amongst these 3 species. Budding is fission is Schizosaccharomyces. Fission is. Then so this is Schizosaccharomyces fission yeast, which is showing vegetative propagation by fission diagrammatic. ही सुद्धा फिगर 2 मार्क्सला विचारली जाऊ शकते, किंवा जास्तच फिगर थोडीशी डिफिकल्ट असेल 4 मार्क्सला सुद्धा विचारली जाऊ शकते. आता इथे इथे क्लिअर दिसते फिजन. दिस इज द काय आहे की सेल आहे, तिथे थोडस ट्रान्सवर्स फॉर्मेशन झालेलं आहे, इथे क्लिअर दिसते, आणि त्याच्यापासून 2 सेल झालेले आहेत फिजन. Then halobile yeast. What is that halobile yeast? Halobile yeast are nothing but yeast with asexual reproduction and intermediate between budding and fission. Budding and fission are called as halobile yeast. That is, they combine these two methods. They combine these two methods. That is what you can say reproduction by both budding and fusion. 2 मार्क्सला विचारलं जाऊ शकतं. Hello Bial is that is the reproduction by both budding and fusion. त्याला hello Bial is असं तुम्ही म्हणू शकता. Hello. आता त्यावेळी सांगितलेलं आहे तुम्ही ते सर्वांनी लक्षात ठेवायचं आहे. 2 मार्क्स 4 मार्क्सला काय विचारलं जाऊ शकते त्यासाठी. Saccharomyces cerevisiae commonly used in bread and beer making. Yeast culture. A culture of yeast can be prepared in the laboratory by placing a piece of yeast cake for few grains of dried yeast in a solution of molasses and water for sugar solution. How to obtain this yeast culture? For that purpose, they have been mentioned. A piece of bread you can place in the laboratory, or dry yeast granules you can suspend in a sugar solution, and you can obtain this yeast culture of Saccharomyces cerevisiae. And obtaining this yeast culture, you can use this yeast culture for various purposes. So yeast cell will multiply rapidly in well-aerated nutrient solution containing sufficient amount of sugar. Millions of yeast cell will be available for study in a day. Within a day, it will be available. The next, thallus. Like other sac fungi, the thallus is a non-mycelial. Entire yeast structure is called as a thallus, and like other fungi, thallus is a non-mycelial. One of the most important characteristics you have to remember. It is not made up of hyphae. There is not a partition. It consists of a single minute oval or spherical cell. It consists of a single minute oval or spherical shape. Some species are elongated. Some of the species are cylindrical in shape. As far as size of the yeast is considered, the size of yeast, which ranges from 2 to 8 micrometer in diameter and 3 to 15 micrometer in length. Each cell appears as a highlight that is colorless in color or transparent in like plant, it consists of tiny mass of a protoplasm surrounding by a cell wall. Like plant, it consists of a tiny mass of protoplasm surrounding by cell wall. If you observe the diagram, you will find Saccharomyces cerevisiae, a diploid yeast cell showing its structure. जे काय आता सांगितलेलं ते तुम्हाला इथे पाहायला मिळेल. Then next is the cell wall. जे काही cell walls आहे, cell wall. So this is a thin, delicate, firm, chitinous in nature. It is made up of 2 polysaccharides. Cell wall is made up of 2 polysaccharides, that is glucans and mannans. These are the polymers of glucose and mannose in combination of traces of proteins, lipid, and chitin, which is a cell wall of the yeast is a thin, delicate, firm, chitinous in nature. It is made up of 2 polysaccharides, glucans and mannan. And glucans and mannans, these are the polymers of glucose and mannose in combination of or along with certain traces of proteins, lipids, and chitin. So electron micrograph of Saccharomyces cerevisiae cell wall shows 2 layers, electron dense layer about 0.5 micrometer thick and inner less dense layer 0.2 micrometer thick containing microfibrils and glucans. तर त्या सेल वॉलची त्यांनी 2 लेयर सांगितलेलं आहे की आउटर आणि इनर लेयर याच्यामध्ये काय फारसं दिसत नाही, परंतु 2 लेयर्स असतात सेल वॉलला. डायग्राम मध्ये परत दाखवलेलं. Then next one is the protoplast and cytoplasm. Protoplast and cytoplasm. Protoplast and cytoplasm. Next to cell wall, that is protoplast and cytoplasm, which is bounded by a cell wall, a cell wall. Protoplast and cytoplasm, which is bounded by a cell. The protoplast and cytoplasm differentiated into two portions. The outer cytoplasmic membrane, which is called as the ectoplasm, and the inner portion of dense or granular endoplasm. The endoplasm and ectoplasm. Ectoplasm. So, the inner portion, dense or granular endoplasm. The endoplasm contains a single small dense nucleus less than 1 micrometer in diameter. 1 micrometer in diameter. This is the nucleus. Associated with nucleus, which occupies much space, is a large vacuole. And you will compare this the space which is occupied in this protoplasm as compared to nucleus by this is the vacuole just space occupied झालेले असताना सांगायचं. The vacuole contains solution or suspension of pollutants, which is a complex material consisting of RNA, which is a complex material consisting of RNA. RNA. The reserve food products in the cytoplasm are in the form of granules of glycogen, oil globules, and protein. Ribosomes, mitochondria are embedded in the cytoplasm. Ribosomes, mitochondria are embedded in the cytoplasm. Ribosomes, mitochondria, these are embedded in the storage, the cytoplasm. Then next, nutrition. As far as nutrition is considered, as yeast cells lack, they are heterotrophic in nature, as they lack this chlorophyll, which is what you can photosynthetic apparatus, photosynthetic equipment. They can obtain their energy, they can obtain their food from this sunlight. Whereas this, in case of yeast, it lacks this chlorophyll. They are heterotrophic. They have to rely, they have to depend on certain ready-made food material which is obtained from the plants. The nutrient elements required are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, magnesium, iron, zinc, manganese, copper, molybdenum. Along with major nutrients, that is carbohydrates, carbon is required, nitrogen is required, and along with this carbon and nitrogen, phosphorus, potassium, sulfur, magnesium, etc. Then, yeah, these are few, these inorganic nutrients, they function as an enzyme activator, acts as a co-enzyme sometimes. Yeast cells secrete zymase, is an enzyme. That enzyme acts on the substrate, and zymase charges complex sugar, starch, or changes, not this is charges, zymase changes complex sugar, starch into simple sugars, and then this simple sugar is used by the yeast cell. The enzyme secretes, and that enzyme acts on this complex polysaccharide, and this complex polysaccharide is converted into sugar. The major portion of sugar is converted into carbon dioxide and ethyl alcohol. This is the glucose. When it reacts with the yeast, it is converted into alcohol, carbon dioxide, and energy. This is the core idea. This is the core point. This is the heart of the what you can say, metabolism of the yeast, that is conversion of glucose into alcohol, carbon dioxide, and energy. Therefore, it is mainly used in various purposes. Various purposes. and the products of this reaction, that is carbon dioxide and ethyl alcohol, diffuse out through the cell wall into the surrounding of this reaction. Carbon dioxide and ethyl alcohol, which diffuse out through the cell wall into the surrounding, and then from surrounding collected. And this process is utilized in the industry. Therefore, there is a great demand for the yeast in the industry for the production of ethyl alcohol. Then next one is the reproduction. जो काय 6 मार्कचा प्रश्न आहे, जास्तीत जास्त मोठा प्रश्न 6 मार्क्सला. त्यामध्ये रिप्रोडक्शन विचारलं जाऊ शकते. मग ते रिप्रोडक्शन फंजायचं असेल, अल्गीचं असेल, ईस्टचं असेल, कोणतेही रिप्रोडक्शन विचारलं जाऊ शकते 6 मार्क्स. फार मोठं असेल तर ते डिव्हाइड करून विचारलं जाईल, आयदर सेक्सुअल, असेक्सुअल वगैरे असं. छोटं असेल तर ते मोठं जनरल क्वेश्चन विल बी आस्क्ड, राइट? Reproduction of the yeast, reproduction of the fungi, reproduction of algae, etc. So yeast reproduces both by sexual means and asexual means. As far as asexual or vegetative method of reproduction is called budding. So that under favorable conditions, Saccharomyces cerevisiae exclusively reproduce by this method, that budding or asexual. So a favorable environment is the presence of nutritive sugar solution. At the time of budding, a small portion of cell wall at or near one pole of each cell softens and thins. Process of budding. The protoplast of this region covered by a thin softened membrane, and that which there is a bulges out in the form of outgrowth or प्रोटूर्बन्स. बल्जेस किंवा त्याला प्रोटूर्बन्स असं म्हटलेलं आहे. दॅट बल्जेस ऑर प्रोटूर्बन्स येत असतं. दॅट इज यू विल फाइंड इन budding, in case of budding. Protuberance, that is a rounded part that sticks from the surface, softened membrane. Then this protuberance gradually increases in size. It is known as bud, and as bud is forming, the nucleus of parent cell divides with its vacuole. So schematically, you will find this schematical diagrammatic representation of this reproduction by budding. Diagrammatic depiction. Budding कसं होत असतं ते इथे तुम्हाला ए बी सी आणि डी मध्ये बडिंग झालेलं दिसतंय. मग अशी मी सांगितल्याप्रमाणे तुम्हाला डायग्रामॅटिक किंवा स्केमॅटिक याच्यासाठी विचारलं जाऊ शकते 2 किंवा 4 मार्क्स. Then next is the fission, vegetative or asexual form. Vegetative reproduction by fission. The division is a transverse. The mother cell elongates, or elongation. The nucleus divides into 2 halves. The daughter nuclei move apart. Then, meanwhile, a ring-like outgrowth appears at the wall of each cell in the middle. This is a ring-like growth appears. Then it grows inward toward the center of the cell. Finally, it stretches across the cell, forming a complete partition. Here you will find a complete partition, which is called as a septum. That is a wall-like structure, septum thickness, and then splits into 2 layers, splits into 2 halls. Not common as budding. It occurs in some yeast and not in Saccharomyces budding is fission. In sexual reproduction, as far as sexual reproduction is considered, it consists namely plasmogamy, that is sexual fusion, karyogamy, that is the union of 2 nuclei, and meiosis, that is a reduction and division. This is a schematic reproduction, schematic depiction of and sexual reproduction. It is halophase and diplophase. Here, 2n chromosome, diploid nucleus is there, haploid nucleus is there. Plasmogamy. At the site with modified cell walls, the two haploid cells of opposite mating types bend towards each other and fuse to form a conjugation bridge. So fusion between the protoplast of plus and minus strain takes place through the conjugation bridge. Conjugation bridge. It is called as a plasmogamy. Then, in case of karyogamy, the two nuclei of the dikaryon fuse, and karyogamy is equivalent to the fertilization. And the diploid fusion nucleus is called as a synkaryon, and the cell containing it is called as a zygote, that is union fertilization of two opposite. Zygotes form as a result of sexual fusions between two haploid yeast cells of opposite mating types or large strain yeast cells. And finally, the diploid nucleus of the spherical ascus mother cell undergoes meiosis, reduction, division. Meiosis 1 produces two haploid nuclei, positive and negative. Each of these divides at meiosis 2 and envelope encloses each of four haploid nuclei together with some cytoplasm. Here it is shown. This is the, in short, sexual reproduction. And lastly, economic importance of yeast. So as far as economic importance of yeast is considered, Saccharomyces cerevisiae, various strains, they are used in the industry because they have got capacity or ability to carry out fermentation of carbohydrate. And during this fermentation of carbohydrate, they ferment these carbohydrates into what we can say this ethyl alcohol carbon dioxide. And that ethyl alcohol has got a lot of significance, importance in various industries. During respiration, the yeast cell oxidizes sugar to form simple organic acid and releases the energy. Various organic acids are also obtained from the using yeast cell. When supply of free oxygen is restricted, the organic acid is split into the carbon dioxide and alcohol. These products have immense value in baking brewing industry. Extensive use of yeast in brewing and baking industry leads to the preparation of yeast cake on commercial scale. These yeast cells are rich in protein, therefore mix with other foodstuffs to increase their food value. That is, all of you are aware, basically what happens, these yeast cells are also used as a, what you can say, as an food for human being, and in microbiological language, we call it as a single cell protein. Single cell protein, and those single cell proteins contain sufficient amount of carbohydrates, proteins, minerals, vitamins in the yeast cell. So fresh yeast cells are excellent sources of vitamin B12. Vitamin B12. So deficiency of various medium is fulfilled by use of these yeast cells. Some yeast have a negative value, that is troublesome parasitic, and classic example is a candidiasis, where candida species which can cause this infection, and the disease is called as a candidiasis. Candidiasis. So economic importance. इतर 2 मार्क्सला विचारलं जाऊ शकते, 4 मार्क्सला विचारलं जाऊ शकते. ते सुद्धा तुम्ही करणं गरजेचं आहे. इकॉनॉमिक इम्पॉर्टन्स. So here we have completed this east also domain eukarya. आपण फंजाय पाहिलं, आपण त्याच्यामध्ये ईस्ट पाहिलं, आपण अल्गी पाहिलं. So if you have any problem, excuse me 1 minute. Yes, if you have any problem.
सर
Yes, कोण आहे? Yes.
प्रोटोझोआ नाही आहे का आपल्याला म्हणजे ह्यावेळी?
आहे सिलेबस मध्ये, पण आपण काय शिकवलं नाही, अजून काय शिकवलं नाही. वेळ मिळाला तर शिकवू आपण. आता आपल्याकडे वेळ फार कमी राहिलाय ना.
ओके
वेळ मिळाला तर शिकवू, शिकवलं तर त्याच्यावरती प्रश्न येतील, नाही शिकवलं तर नाही येणार प्रश्न. कोण आहे? कोण कोण बोलतंय? येस,
प्रज्ञा सर.
प्रज्ञा. वेळ मिळाला तर आता पुढच्या आठवड्यात परत FYBC च्या एक्झाम्स आहेत, त्यामुळे काय, आणि तुमच्याही एक्झाम आल्या ना आता. तुम्ही जे काही 16 युनिट्स आहेत त्यापैकी कुठल्या युनिट मधलं काय राहिलंय काय नाही ते त्या टीचर कडून हे करून घ्या. आणि तुमच्याकडे सगळ्या हार्ड कॉपीज पाहिजे. तर हार्ड कॉपीसाठी ज्या काय या PPT आहे त्याच्यापेक्षा ओरिजिनल कुठे काय जे काय पब्लिश सायंटिफिक लिटरेचर आहे ते कलेक्ट करा आणि ते वाचा. देन यू विल बी एबल टू रिप्रोड्यूस इन द एक्झामिनेशन. म्हणून काय शंका आहे का? इज देअर एनी डाऊट? आज किती वाजता बोलवले कॉलेजला तुम्हाला? येस,
12 वाजता.
12 वाजता बोलवलेलं. त्याच्यानंतर काय शेड्यूल काय मला माहिती नाहीये. यू कॅन आस्क द शेड्यूल टू युअर रेस्पेक्टिव्ह फॅकल्टी. ते पवार सर किंवा काय कोण घेणार आहेत काय ते बघायचं. नाव आय वुड लाईक टू स्टॉप हिअर प्रेझेंट अगेन.